Good afternoon. This is an EMBL-EBI Resources and Introduction webinar. My name's Tom Hancox. I'm a Scientific Training Officer here at EMBL-EBI. My email address is tom@ebi.ac.uk. If you want to contact me, please do so regarding this webinar. I'm happy to send out slides afterwards as well. So this webinar should take about half an hour. I'm just going to run through a little bit about EMBL-EBI itself, the work we do here, and also the various bioinformatics resources that are available through our website. For those people who are not aware, EMBL-EBI is part of the European Molecular Biology Laboratory. We are based here in Hinxton in the UK. Our headquarters is in Heidelberg in Germany. There are also structural biology outstations in Hamburg and Grenoble, and a mouse biology research unit in Monte Rotondo outside of Rome. EMBL employs over 1,800 staff across 60 nationalities. So wherever you are at EMBL, you have a very diverse community of people working both in sort of research, in technical services, and in other support roles. EMBL itself is made up of a number of member states, obviously initially from across Europe, but also including associate members Argentina and Australia. And over the years as well, we've grown in size with current prospective member states, Hungary, Poland, and the Slovak Republic. So EMBL-EBI itself is an outstation of EMBL. We are the European Bioinformatics Institute. We are a not-for-profit research organisation, and our main role is to act as a hub for biological data across Europe. As part of this, we offer numerous services in bioinformatics, data storage, submission and analysis, as well as conducting world-class research into bioinformatics, computational biology. So our main mission is to provide these bioinformatics services, to conduct research that will drive forward the community that we work in. We provide training, webinars, and also face-to-face training with PhD and postdoc students. We also have a strong industry program and act to coordinate data provision across Europe and with others around the world. So, as I have said, this webinar is focusing on the bioinformatics services available at EBI. These cover the entire spectrum of modern molecular biology, ranging from genes right down at the DNA level, looking at the variants, variation across populations, looking at how population alleles shift and the different phenotypes associated with these gene variants. We look then at sort of the level of expression, so be that genes and proteins, or even sort of small molecule levels, metabolites within body tissues and samples. We also have resources that cover protein sequences, the different families and motifs that those protein sequences can form. We have resources that look at molecular structures, so looking in 3 dimensions at the protein sequences and how they form 3-dimensional proteins that interact with other proteins and DNA. And that can be electron microscopy data, magnetic resonance imaging, and a whole host of novel techniques. We also have resources in chemical biology, looking at bioactivity and different compounds. Then we move into the level of reactions, interactions, and pathways, where we look at how these different biological entities interact with each other, form networks within models, and of course beyond that to the whole system, ecosystem sort of level, looking at different ways of organizing and understanding information. And then finally we move into the literature and ontologies area, where we look at obviously the scientific literature, how we can link that with our own data, but also novel ways of organizing, maintaining and gaining extensive knowledge from data that we hold. So the research at EMBL-EBI covers a range of different biological areas, the genetic, the expression, transcriptomics, metabolomics, and protein expression, looking at protein sequences and structures, the biology behind those, looking in the chemical biology domain, and also into pathways and systems biology. The training we offer at EMBL-EBI comes in several forms, offering training on site here on campus in Hinxton. We also offer off-site training, sending dedicated trainers to organisations around the world. And we also offer training online through webinars like this, and also through a dedicated e-learning portal called Train Online. EBI's industry programme involves partnerships with a number of companies in the pharmaceutical, chemical, agrochemical and consumer goods areas. We work with them to allow them to access the data that we hold, work on special projects, and to help disseminate the information and understanding to industry across Europe and the rest of the world. So, what do we really do? So, EBI is very much like a very large data repository. We have over 11 million requests submitted to our website every week. That's people catching up on news, searching for stories, searching for genetic information, looking at protein sequences, and we estimate that over 5 million unique visits are made to EMBL-EBI websites. So that could be 5 million scientists in the UK, across Europe and around the world. And our clusters and servers handle over 9.2 million jobs on average per month. So that could be very simple queries through our search engines, or more complex sort of data handling exercises running large jobs on our servers. And in total, EBI has over 60 petabytes of data storage capacity. So meaning that scientists from around the world have deposited large amounts of biological data with us. So back in 1984, this is a picture of the nucleotide sequences database, or a copy of it, available part 1 as a fairly hefty book. So that was over 32 years ago now, available in print form, some 500,000 DNA sequences within the data storage at what was EMBL Bank. However, times have changed. As I'm sure you're all aware, there are new technologies out there, genome sequencing, proteomics, mass spectrometry, and a whole host of other large-scale, high-throughput technologies that biological, chemical scientists across the world are generating vast amounts of data. As part of the publication and submission process, they have been depositing data at EBI. So this graph here shows from 1996 to 2012 an absolute explosion in the data that we held at EBI. Very difficult to see here on the graph, but between 1996 and 2006 we have an exponential growth in that data, and then again in the 6 years from 2006 to 2012, again that just keeps on growing. So just to put the amount of data that we hold in perspective, in 1996 equivalent to 200 gigabytes. By 2006 that had grown to 238 terabytes, and in 2012 that was 16 petabytes. 1996, that is the equivalent of 40 DVDs of data, perhaps the size of a large tablet these days. 238 terabytes is the equivalent of over 250 years of government proceedings from the US Library of Congress. And that 16 petabytes, really to put it in the scale of some of the other sciences out there, the 16 petabytes of data we held in 2012 was the equivalent of the Large Hadron Collider at CERN conducting particle collision experiments for just 1 year. So whilst biologists are going through an absolute data explosion, we have a very long way before we're catching up with high energy physicists and their incredibly large machines. So, as I've just mentioned, scientists around the world, as part of your publications, you will be depositing data with us. That could be DNA sequencing, genomes, it could be metabolic profiles, chemical screening data, it could be protein sequences or the structure of those proteins. They get submitted as part of a publication that you'll be making in Nature or Science, and we archive those away in large repositories where they're available for people to access. We also do a lot of additional work as well. We classify all the different data that we get submitted. We put it into new databases, we move it around, we pair it up with other data, and we're able to share it back with you. So all of this data that EBI holds is freely available, freely accessible. So you can search through, as we'll have a look later on, and see the different types of data that you can get hold of. We'll provide ways for you to analyze it, different tools that, following our own analysis of the data we have, we can provide new tools, new services, new resources on top of this data, and obviously share it back with you where you can take that data and go on and learn new things, discover new things, and publish more in the future. Whilst we are the European Bioinformatics Institute, it's important to stress that our users are worldwide. Here is a very short glimpse of just some of the users accessing the EBI website, and of course this will change during different times of the day. We also have very strong collaborations with researchers in Japan and in the United States, sharing the data that the NCBI and the DDBJ holds, allowing you to sort of access mirrored information at any time or anywhere in the world. So, obviously I've just explained that we have this wealth of information. I'm now going to take you through some slides just briefly explaining where you can start out to explore and find this information, and then go about accessing it and making use of it yourself. So I've already gone through the services that very briefly are available at EBI. We'll be taking a little bit of a closer look, but do remember, sort of as we go through, I'm only able to show you a very brief snapshot of the services we provide. I encourage you to sort of explore the EBI website, have a closer look, see the different things that are available. If there are services that you are interested in, please ask in the chat window. My colleague Laura will be able to assist you, hopefully directing you to where you may be able to find the things you're looking for, but also please check out the webinars as well that we'll display later on at the end of this session, that will give you some idea of some of the upcoming things you can learn about through our webinar series. So it's important as well to stress that each of the databases, the resources, the tools and the software at EBI are not independent entities. There is a lot of work that we've put into to connect all these different resources, allowing you as a scientist, as a data analyst, to work through different websites, following different trails of data. So we may, for instance, be interested in a particular part of a genome. We can look at that genome in the European Nucleotide Archive, the ENA. We may be able to then follow that gene sequence and look at the translated protein sequence, and we would find that within UniProt. From UniProt, we have many different ways in which we can link out to other data, maybe looking at expression information, looking at the context of that protein on a genomic scale, where the different isoforms are found, the different introns and exons in the gene that encodes that protein. But of course, we can also look at the three-dimensional structure that is formed from that protein sequence. From there, perhaps we can go and look at different chemical biology resources. For those of you interested in drug discovery, you may want to know sort of your protein of interest, if it's a potential drug target, if other people have already conducted experiments that show that your particular protein interacts very strongly or doesn't at all with a compound, small molecule of interest. And of course, all of our resources are linked through to Europe PubMed Central, collaboration across Europe that brings together literature services, allowing you to access literature, free text articles, free text abstracts, but also linking back from those literature articles, the citations, back to relevant data and information at EBI. So finding a publication on a gene of interest, you should be able to go and find the gene sequence of that gene of interest from the article. So the European Bioinformatics Institute, our website, we are www.ebi.ac.uk. Laura will post that in the chat window now. If you want to go to that website, I'll do a very brief walkthrough, just orientate you around the website if you're not already familiar with it, point out where you can find the different services, different tools, and where you can obviously find out more information. So along the top there are several tabs, the services, research, training, and about us tabs. There are also several connections as well to the services, the research, training, industry, the European coordination roles, and also alumni resources. Further down the page you'll find interesting news articles, some of the projects that EBI researchers are involved in, new publications that have come out from EBI staff as well, other information as well about visiting EMBL and its outstations as well. So if we were to click on either the services button or the services tab on the top, we get taken to the EBI services page. This is an overview, again based on that biological spectrum, the arrow that we showed you before, breaking our resources down into different areas, which hopefully should be quite intuitive, easy to understand where you may find resources of interest. Obviously DNA, if you're wanting to look at genes, genome and variation information, right through to sort of chemogenomics, metabolomics and chemical biology. And of course, if you're looking at ways of storing data, relating data to one another, you may be interested in the ontology resources. Of course, the literature services I've just mentioned, and then a whole host of cross-domain tools, software, and other ancillary databases. Further down that page as well, you will find information on programmatic access. So for those of you who work in bioinformatics, who are actually computer scientists, you may be interested in accessing a lot of the EBI data resources through our API. You can look, follow that link that will take you to more information as well. And also if you're an EMBL-EBI collaborator, there's further information that you can gain there on the Embassy Cloud, which allows you to move your tools and services very close to our data, allowing you to work alongside the bulk of our data without having to transfer that to your own institutes. So if you were to click into one of these service domains, here I've clicked on the DNA and RNA field, you will see there's a very long list of the most popular services. Obviously here Ensemble is our top access DNA resource, where you can browse a whole number of vertebrate genomes, looking at obviously the DNA sequence right up to the chromosomal level. Ensemble Genomes provides a virtually identical source of data and information, but for non-vertebrate species. And then we also come to our first item of sort of our tool or item of software, which is Clustable Omega. Hopefully you're familiar with this multiple sequence alignment tool, which allows you to align DNA and protein sequences. We have other resources as well. One of our main archival resources further down, the European Nucleotide Archive, and then beyond that, 1,000 Genomes, a project which catalogued human variation across the world. So a very brief snapshot there. You can drop into any of the other domains to see the different resources that are available. I'm going to move on now, but please feel free to come back in future, have an explore of those. I'm now going to outline EBI Search. So this is a search engine integrated within the EBI website, which should allow you fast and quick access to relevant information and data. That is the large search box that you'll find in the center of the home page. If you are browsing around other web pages on the EBI, you'll often find another search box appears in the top right hand corner of the web page. It is just the same interface, and just like internet search engines, you can type in genes, proteins, chemicals of interest. You can also obviously type in tools and resources as well. So for this example, I'm going to search for BRCA1, well-known gene, protein involved in breast cancer. So typing that into the search window, if you were to hit enter for a short wait, you'll have a return page, hopefully very, very similar to this, which outlines a large amount of summary information in a number of filtered results. You can obviously look at genome information here, so this will include multiple organisms that have had their genome sequenced and which BRCA1 analog has been identified. Through nucleotide sequences, potentially mutant sequences that could have been implicated in breast cancers or other organisms where similar mutations have been found. Obviously the protein and macromolecule information relating to the BRCA1 protein. Small molecule information, so potential drugs that may be implicated in sort of maybe the treatment of BRCA1 or other small molecules that might interact with the protein or its complex. Information on gene expression, so this could be RNA or microarray experiments detailing both temporal, spatial and disease-specific expression of the BRCA1 protein. Information on the molecular interactions that the BRCA1 protein undergoes, so that might be with DNA, with RNA, with other proteins, obviously large complexes or individual small proteins, and of course the molecules it might interact with. And obviously further information, probably from a host of different organisms, on the reaction pathways that the BRCA1 protein is involved in, those upstream and downstream proteins that it interacts and works within the human cell, and of course disease information as well. Protein family information as well, where you can learn more about related proteins to BRCA1. That may be genes, proteins with a very similar function, or those that over evolutionary time have undergone evolution and changed into different forms and functions. Information on the enzymes that may interact with BRCA1, information and citations in the literature, so over 10,000 citations in Europe PubMed Central, and other information as well on how it's appeared in different ontologies and also in biosample experiments and systems biology information. There is also at the very bottom there as well just the number of returns as well on the EBI website. That may well be web pages that detail the BRCA1 protein in news articles. Maybe we've published papers here at EBI that have been advertised as well on the website. Over in the center as well, you will find this page will scroll down and down. It will return all the different search results I've just talked about. But importantly as well, staff here at the EBI curate these gene and protein summaries, which basically pull together the most useful and relevant information that we found our users want to find out or continually access at EBI. So providing several model organisms that BRCA1 is present in, and if you were to click on one of those, you would be taken through to that particular species summary page, which I'll do now. And you end up with some collated information in several different areas. Obviously, starting at the gene level, we can look at the sequence of this gene using some of the DNA and RNA resources I highlighted earlier, those being ensemble repositories where you can download that sequence, and obviously view it in a genomic context. Here you'll see there is information on the number of transcripts in the entire gene, and the number of exons, the size of the transcripts as well. More information can obviously be found by jumping out and following these links. You're also able to access other information using the tabs down the side, so looking at expression level, using some of the Array Express and Expression Atlas resources, providing body maps showing where and when the BRCA1 gene is expressed, if it's upregulated or downregulated in different conditions. You can move down as well to find more protein information, looking in sort of UniProt, the protein sequence itself, the different interactions that protein has and intact, looking at resources like PFAM, which will provide information on similar proteins in the similar families to BRCA1, looking at PDBE, Protein Data Bank in Europe database that contains structural information, so atomic coordinates of the individual atoms in the BRCA1 protein, but also potentially information on sort of electron microscopy mapping as well. And again as well, you can access the literature services here at EBI to find citations of BRCA1, papers of relevance and interest in the literature. So just coming now towards the end of this webinar, hopefully that's giving you a very brief introduction to the EBI website, where you may be able to find more information. I also encourage you to sort of get in contact with us, both myself, the email address I provided earlier, but also if you're on social media, the EBI training team will be tweeting about upcoming courses, events, and of course webinars. So you can follow us at EBI Training on Twitter. EMBL-EBI itself has its own Twitter account where we post information on sort of exciting research projects, new publications, conferences that EBI staff will be attending, different events as well around campus and other places around the world. There is also a list as well that's been put together of EBI staff members who are active on Twitter. Maybe interesting if you want to follow some of the scientists here at EBI who engage in sort of social media, learning about what they're talking about, the papers they're reading, the exciting things that they're learning, and people they're meeting. Also as well, if you ever attend courses and events at EBI, we update our Facebook page with some exciting information just about the courses and the events that have gone on. And of course, not only does EBI itself engage with social media, you'll find many of the different resources at EBI have Twitter and Facebook pages. PDBE often shares exciting and interesting protein structures every week. On Facebook, on Twitter, you'll find updates and information from the different resources. If websites go down, if there's maintenance occurring, they'll provide updates as well. Just to let you know and keep you informed of when services may go down. So finally as well, I'd like to make a few acknowledgements, in particular to my colleague Laura Emery, who sat here next to me, who's been answering your questions on the chat window, and the rest of the training team here at EBI. So if you ever come to any of our courses, if you ever attend any workshops, you may meet us in future, please do get in contact with us. You'll also see over on the right-hand side our different funders. So EMBL itself, the European Molecular Biology Organization, EMBO, the European Union, European Commission, a number of European projects such as the IMI project, the BBSRC funding body here in the UK, and of course the Wellcome Trust, who not only fund us but provide the wonderful campus that we're situated on here in Hingston.